get from here? I always feel that when I hear talks like yours, and actually my PhD is in ecology, and at Princeton, so I know Steve Call, is that it all comes down to psychology. So in some ways, even though I've gone from ecology to psychology, that's where it all comes down. If we can't change human behavior, it doesn't matter how much technology we have. So that's what I'm going to address today. Where did learning come from? Where did the brain come from? And really getting back down to first principles. I also want to just note, speaking of innovation, that today is both Charles Darwin and Abraham Lincoln's birthday. So here's a good day for innovation, a bad day for anti-abolitionist Christian fundamentalists. Bad day for the South in general. Okay, so I want to talk about how the brain evolved from a nose, and because last time I went over, I'm hoping to be right on time, so there's going to be a fairly data-free talk. So what do we know about the first brain? And this is actually very interesting. We know nothing about the first brain. The history of animal life was that for millions of years, there was no fossil record of multicellular animals except for little tiny worms living in algal mats. And then suddenly, in the Cambrian Explosion, 40 million years later, we had 100 animal phyla, most of which we still have, and no new phyla have evolved since then. So it really was a big bang of evolution, and really a cognitive big bang, because before that, there was no evidence for neurons or brains, and afterwards, this is a beautiful John Civic's painting from the Burgess Shale, and by the end of this period, there were all these sensory systems that modern invertebrates have now were pretty much present. Complex compound eyes, olfactory antenna, mechanosensory hairs, et cetera, et cetera. So the problem is we have no fossil record. So we don't know why suddenly there was no brain and then there was a brain. So we can, a hint we have is that we were underwater. And that's important because that's going to limit what was there. And the thing about being underwater is that it's a chemical world. And chemicals are very interesting. Olfaction, of course, is the sensory modality that encodes information using chemicals. Number one, it was the first. All living animals, single-celled bacteria, everything uses chemicals to communicate, to decode their environment, to decide what's out there, how to react. It's also not only the first, but it's the most universal. Yes?
Can you put the mic closer to you?
Oh, close to my mouth. Okay, sorry. Okay, so thank you. Chomp. I also study squirrels, so I might chew on it. So it's universal. So it's the use of chemicals to learn about your environment is found in all animals. It's also the largest investment. If you look at the size of the proportion of your genome devoted to any one function, not just in mammals, but also in fruit flies, also in nematodes, the largest gene families are devoted to olfaction, olfactory receptor gene family. So it's clearly not just an accident that we used to use it. We're still pumping in a huge amount of energy to maintain these systems in our bodies. And what I've been thinking about is that we think of olfaction as something that just is you use to identify what's out there and categorize it. I've been thinking about it differently. I started thinking about how the brain evolved under the water and realized that the only system, if you want to map large areas of space, the only way you can do it is by using olfaction. It's the only sensory modality that has any kind of ability to encode remotely, picking up a gradient of odors on the water. So another civic painting of the Cambrian Revolution. Most of these animals are no longer with us. So what caused this revolution? What was this cognitive snowball that began in the Cambrian? It was the ability, the first time that animals moved beyond just reacting, but to actually to learn and to be able to associate stimuli together. Now, why would you again, getting back to first principles, why learn? Well, if you can learn, then you can predict. You can predict if you've got an association between this and this stimulus, then you can, if you're looking for stimulus B, you can then now look for stimulus A. And that allows you, for the first time, to control your environment. And that's, of course, what we're all about. Bacteria to humans, we're all about homeostasis and knowing where the things are that we need. But to do that, you need to map space. So what I've argued is that this Cambrian explosion occurred when the first animals harnessed the ability of olfaction and were able to create maps using chemicals and were therefore able to outcompete and become these ferocious predators. The Cambrians known as the bloodbath. So what about mapping and navigating? Actually, it was in 1949 that two scientists were the first ones to study and demonstrate that animals map space. And the first one was Edward Tolman at Berkeley, who coined the term cognitive map, studying rats in mazes. But at the same time, and I don't think there was no evidence that they knew of each other's work or corresponded. Gustav Kramer in Germany was studying the homing pigeon, which of course had been domesticated by thousands of years earlier as a messaging system, but he was studying how the amazing feat of a homing pigeon, which is you can release it 300 kilometers in an area it's never been before, and it can beeline back to its loft. And he was using, these two men were both studying, and for the first time, these two scientists said, you know, these animals are not stimulus response machines. This is not Skinnerian behaviorism. These are animals with representations who actually have created a map, a cognitive map in Tolman's language and the map and compass model of Kramer. And recently, there's been a lovely work in the Egyptian fruit bat, which has replicated for the first time in a mammal, of course it has to be a flying mammal, the work of the pigeon by releasing these bats, wild bats, in the Negev desert in a crater where they can see no landmarks. They're released in the and they and just this is the GPS log, and they circle just like a pigeon, come out of the crater and beeline back to their log. So how do they do that? How are these animals navigating without landmarks? How the pigeons are doing it, and now we've got a mammal doing it. This is going to tell you how they can use olfaction. This is how do you do that? How do you map without landmarks? Well, what you need is vectors, and because you want to create a grid from vectors. So say you start off with gradients such as odor, sound, magnetic fields. You can also have features, physical features of the landscape, the slope, or a vegetation, a permanent vegetation line, or rivers, roads. Homing pigeons are known to use highways to navigate now, follow highways. So you can use, you can have these simple vector from just looking at the slope of a mountain versus the river, and then you get you can extract the vectors from that, and then simply make a Cartesian grid with that. And that's what allows you to localize objects, localize a goal in the absence of landmarks. And this is something that I've been working on for a while because I was very interested in how how navigation evolved. That's my background is evolutionary biology, and I got interested in how cognition evolves, and I've been focusing on spatial navigation, again, because it is the universal cognitive ability, and so you can look at how it evolved. And this particular navigation is particularly interesting because it's encoded by the hippocampus, which is, there's your hippocampus, but these are coronal brain slices of a rat, turtle, chicken, lizard, and all five of these species, the hippocampus does the same thing. The hippocampus is homologous. It's found in all vertebrates. It is one of the most ancient of all brain structures, and it's huge in most. Yours is pretty big. You should see the rats. And the rat and all these others. It's always a very large structure. And the last fall, the Nobel Prize in Medicine went to these wonderful John O'Keefe and the Mosers for their work showing how the hippocampus encodes space. So we know the hippocampus encodes space. We know the animals all encode. We're done, right? Well, we're not done because of the work by Ken Lohman and Randolph Menzel showing that animals without hippocampi, the honeybee and the spiny lobster, can show exactly the same kind of mapping. So this is one of the paradoxes that us in the field are left with. I've developed models of vertebrate hippocampus, but then I'm left high and dry when it comes to invertebrates. And so what I started thinking about is, what is the common denominator between the honeybee, the crustacean, and all the vertebrates? And what did they have in common back in the Cambrian? And that's actually olfaction. And in fact, it's not just primitive animals that use olfaction. Homing pigeons, who are extremely visual, much more than we are, rely more heavily on olfaction than any other sensory modality. There's 40 years of work now showing this. And so this is a homing pigeon, and these are 4 birds that are normal, and they're all about 300 kilometers from the center of that circle, which is their loft. And over here, you've got 4 birds whose olfactory abilities have been blocked or lesioned in some way, and you can see they're very disoriented, even though they have their vision is unaffected. That is what happens when you take away their nose, and that's an extremely, visual animal. So it turns out that your nose and the honeybee's nose and the crustacean's nose, we actually have there's similarities. We're organized in these little balls of neural processes called glomeruli, and those glomeruli are found in all animals, including mollusks and insects and vertebrates, and except for one, the C. elegans, lacks it. And not only this is a honeybee brain, and it actually looks different, but it's actually organized. The neural circuitry is exactly the same as the neural circuitry of your nose. Its nose and your nose, and the same neural codes and oscillation patterns that are used to decode it are the same. The same psychophysics. There's all these enormous parallels that have been discussed for decades, because people who study honeybees can study the same neural processes as someone who's studying a human nose. And recently, Detlef Arendt's group shown that the brain of this is the kind of best equivalent we have for the Cambrian, which is an annelid worm. And if you look at the sequence of genes that encode that its brain during its forebrain during development, it's exactly a homologous sequence of genes that encodes the forebrain of the mouse. The forebrain is the location of the hippocampus. So what we've got is their suggestion that there's this convergent evidence that there really was a Big Bang. There was, and during the Cambrian, one brain appeared, and this one brain is encoded using the same homologous sequence. And then, of course, since the Cambrian, we've had wild divergence, but both the genes and the function may still be homologous. And so that's what I've working on now, and I'm not going to show again the data, but I've been proposing that the Cambrian Big Bang was the use of olfaction to create maps using what is in what I call a parallel map architecture, which I don't have time to discuss, but is seen both in it's basically the Cartesian grid without landmarks is what I showed you earlier, and the evidence for it, if you look at the size of the olfactory bulb in carnivores, for example, where it's the size of the olfactory bulb in birds and mammals and dinosaurs and insects, is not predicted by their diet breadth or how many different kinds of foods they eat, but actually by how much space they use. And in fact, after I published the paper in 2012, new analyses have come out showing in primates even, you've got olfactory bulb size is co-varies with home range size in wild monkeys, which no one had ever looked for because no one had ever expected that. So you've got olfactory bulb and hippocampal size and home range size all co-varying. So what I'm arguing is that we have to really think differently about olfaction. To be completely extreme, I would say the primary function of olfaction is navigation. Your nose is a navigational device. Secondarily, you use it to discriminate things. But if you ask across metazoan animals, what is the primary function of the nose, it appears to be olfaction. Olfaction explains more variance in the size of the structure, the processing of the structure, how the psychophysics works. It's really quite surprising. It's not something we'd really thought about before. So I'm calling this the Proust hypothesis for the reason I'm calling, because it's actually, in terms of vertebrates, it's really, what it really is the evolution of the limbic system, because the Proust is predicting and representing odor utility in space and time. That was, you can tell I spent months trying to get that to fit into Proust, but I finally did. And it's the where the olfacto-hippocampal system, which is together, the olfactory bulb and the hippocampus are creating a map of what is where, and then I haven't talked about the amygdala, but the amygdala is just as important, and in fact, it's just as ancient, and the connections and the size and the plasticity of these three structures are linked. I mean, you can't really separate them. And in fact, again, something else I haven't had time to talk about, but it turns out that you think about the brain, you never get new neurons in the brain. Well, there's one place that's fairly well known now. The hippocampus is the one place in the vertebrate brain where you can get new neurons, and the olfactory bulb, it turns out, is the second place. And in fact, the olfactory bulb and the hippocampus are the only two structures in the brain of all vertebrates across the board that have adult neurogenesis. And it turns out that the medial amygdala, which is the part of the amygdala that is most closely cued into the olfaction, also has adult neurogenesis. Though not to the same degree as the olfactory bulb and the hippocampus. So you really have a very tight system. In fact, the hippocampus used to be called the rhinencephalon, the nose brain, because before John O'Keefe, we didn't know what it did, and the only thing it did is something with olfaction. So I think I'm just coming back to that with my Proust hypothesis, and it's not completely silly, because Proustian memory, his memory of the Madeleine dipped in linden tea, was a very interesting memory, because what it was taking a primitive, a very primitive sensory input of a smell, basically, because most of your taste is actually olfaction, and with it he was being suddenly recalled a very, very specific spatial and temporal memory. In fact, it was a episodic memory, which is considered to be many people feel it's the only unique form of memory in humans. It's certainly a very complex memory that creates all your autobiographical memories, and yet it's stimulated more by olfactory cues than any other sensory modality. And I would say the link between space, time, and olfaction, which is Proustian, is because the brain began that way, and olfaction is the architecture. The basic archetype of the brain and cognition is olfactory spatial mapping. Thank you very much. Good job. We started it all.